Hi and welcome back to Equity Tutors. Today I'm going to be starting the exchange module. So this is going to go over a couple of weeks, and what I'm going to do today is give a brief introduction to some concepts that you're going to need later on in the module, and kind of a brief overview of what exchange is and why we need it. And then in the following weeks, Amelia will be going over exchange in animals, and I will then be going over exchange in plants. So to get started today, I'm just going to talk a bit about membranes. So you'll remember from the previous modules on cells that membranes surround the cell and they allow control of what can come into and out of a cell. You will have heard me before refer to membranes as a phospholipid bilayer, and you'll remember that from the cells module. So phospholipid bilayer means that it has the bi means that there are two layers, and the phospholipid, which are the individual molecules that make up the membrane. So going a bit deeper into exactly what the membrane is, we're going to talk about the fluid mosaic model. So in 1972, this model was described, and it explains how biological molecules are arranged to form the cell membrane. And this is a phospholipid bilayer, as I previously said, but it also contains other molecules that are not these phospholipids, and I'm going to go into that in a bit more detail now. So the phospholipid bilayer is composed of two layers of phospholipids, and if you'll remember from our first module, they have a hydrophobic tail, which means that it doesn't like water. So these face inwards, and then you have a hydrophilic head, which faces outwards. So what you get is you get these two layers, and you get both lots of the tails in the middle and the two heads are on the outside. This will all make a lot more sense if you go and have a look at the YouTube video, or if you have a look at the learning resources that I've attached to the bottom of the podcast. So there are three main things that make up the membrane. So we have phospholipids, which I've just talked about. We then have cholesterol, which is found in animal cell membranes, and this provides stability. And we then also have other proteins, and these can be integral, which means that they span across the entire membrane. So these are things like channel proteins or carrier proteins. Don't worry too much about that right now. We're going to go into that in a lot more depth throughout the lesson. And then we also have other things that are called peripheral proteins, such as glycoproteins, which decorate the outside or the inside of the membrane. So they don't span the entire membrane, but they poke out from either side. And the membrane is very important for not only controlling what goes in and out of the cell, but also for cell signaling, so communicating with other cells and things like that. And this is where these other peripheral proteins come in very useful. So because the phospholipid bilayer is made up of these phospholipids, the phospholipid bilayer is actually fluid, so it's very viscous, but within it, every individual phospholipid can actually move positions. We then also have these other proteins that I've just described, and they cannot move as easily. And what happens is certain molecules, like cholesterol, fit in between the tails of these phospholipids, and this provides stability, so it stops the phospholipids being able to move around quite so easily. And then when we're talking about the fluid mosaic model, where those words come from is fluid because there is movement within the membrane, but also the mosaic is because it's not just made up of one molecule, it's not just made up of phospholipids, it's also decorated with these other proteins, these channel proteins, carrier proteins, peripheral proteins, cholesterol, all of these things, which we call a mosaic because it's like a mosaic you would see has lots of different parts to it. It's not all just one thing. Going on from this, we're then going to talk about a bit more about the structure of the membrane and what's within it, and what the uses of these different proteins are. So we can broadly break down the phospholipid bilayer into containing three types of lipids and two types of proteins. So we have phospholipids, cholesterols, and glycolipids. And you'll remember these words from the lipids lesson, the lipids and carbohydrates lesson in the first module. And then we can also have two different types of proteins. So we can have glycoproteins and then other types of proteins as well, and these are normally the ones that are transport proteins. So as I mentioned before, transmembrane proteins span the entire membrane, so they go from one side to the other, and we can also call these integral proteins. But peripheral proteins protrude from either side, they don't run right through the membrane. So an important thing that the membrane does is controls the movement of substances in and out of the cell. However, certain substances can move easily through the phospholipid bilayer. For example, non-charged small molecules can move easily between phospholipids. However, other molecules cannot move easily through the phospholipid bilayer. And the cell needs to bring molecules in, for example, glucose and water, but it also needs to get things out of the cell. Waste products need to be taken out of the cell. So the way that this is done, not everything can move through the membrane, as we said, so we have these transport proteins. Now, transport proteins create a hydrophilic channel through the membrane, and this means that ions and polar molecules can travel through from either inside to outside or outside to inside, and we can further break these transport proteins down into two further subtypes. So we have channel proteins and we have carrier proteins, and I'm going to go into more detail on those two in a minute. So each transport protein is very specific to whatever molecule it is trying to move in and out of the cell. So that's very important to remember, that not all transport proteins move the same things. They're very specific for whatever molecule you are moving in and out. And transport proteins really allow the cell to control what goes in and out of the cell, which is very important. I'm now going to go over how substances can move in and out of the cell with reference to these transport proteins, but also via diffusion and things like that. So I'm just going to do a brief overview of diffusion again. We did go over this in one of the early lessons, but it's always good to go over it again. So diffusion is the net passive movement of molecules or particles from regions of higher concentration to regions of lower concentration. So this is a really important definition to have in your brain. You really want to add this to your glossary because it will come up time and time again. And in this module specifically, it's going to be your bread and butter. So diffusion is the net passive movement. So the important thing here is that it's passive. So this means it doesn't use energy. So what this means is that if you just leave some molecules, they will, on their own, move from areas of high concentration to areas of low concentration. We can think about this by imagining someone spraying deodorant. If you're close to the deodorant being sprayed, you will get a high concentration of it. But then eventually it will even out and everyone in the room will be able to smell the same amount of deodorant because it will have moved from that area of high concentration and it will just spread out until everywhere has the same concentration, and it will all be lower. It will have moved to the areas of lower concentration. There are several things that can affect the speed of diffusion. So first of all, the steepness of the concentration gradient. So the higher the difference between the high regions and the low regions, the faster diffusion will happen. If you have two things that are quite similar in concentration, that diffusion will take much longer to happen. We also have temperature. So the higher the temperature, the faster the diffusion happens because the molecules within that substance will have more energy and will be moving more quickly. So if you have a gas that is very hot, it will diffuse much quicker than a gas that is cold because within it, the particles have much more energy and are moving around much more rapidly. So they will move from the area of high concentration to the area of low concentration much more quickly. We then have surface area of a membrane. So if things are diffusing through a membrane, if they have a larger surface area to diffuse through, that's going to happen quicker. Imagine, you're queuing up to get into a shop. If there's lots of people, it takes longer for you all to get through the one door. But if there are 3 doors, you'd all get in quicker. So surface area can also affect the rate of diffusion. We then have the size of a molecule. So if the substance that's diffusing has very large molecules, then that will take longer because they will require more energy to move. And also we have distance. So you will have a higher rate of diffusion over a shorter distance than over a longer distance because it will take the particles longer to reach the areas of low concentration if the distance is greater. So hopefully that all makes sense and is kind of a bit of a revision on what we'd covered before. But also, hopefully this also makes sense in your day-to-day life. You know, you can think of this very clearly in situations you encounter every day. So we see diffusion happening all the time. So a specific example of diffusion is osmosis. And again, Amelia covered this in one of the early lessons on water. But just to go over it again, osmosis is the net movement of water molecules from a region of higher water potential to a region of lower water potential through a partially permeable membrane. Now, I know you're probably sick of us saying this, but a really important thing here is to know the words partially permeable membrane. So osmosis has to occur through a membrane that doesn't allow everything through, and that is what partially permeable means. Osmosis all depends on the concentration of water molecules within a solution. So this is affected by the concentration of anything else in the solution. So if there's more sugar in water, then the water potential of that glass of sugar water is lower than a glass of pure water because there is another solute in that water. So when we're describing solutions, we're normally talking about whether the concentration of water is higher inside or outside of a cell. And this is really important because the water potential of a cell is what allows it to keep its shape and either not become shriveled or also not explode and burst. So the way we can describe this is hypertonic solutions, so ER, is when the outside of the cell has a higher solution concentration than inside the cell. So that means that there's more water inside the cell than outside, so the water will move out of the cells to even out that water potential and make each side equal. We can then say that if the outside of the cell has a lower solute concentration, so there's more water outside the cell than in the cell, then we call this a hypotonic solution. So that's with an O. So hypo and hyper. So with a hypotonic solution, as the higher concentration of water is outside the cell, you're going to get water moving into the cell, and that can cause some issues as well. So I'm going to go on to that in a second. And then if there is an equal amount of water inside and outside the cell, you will get no net movement of water, and we call this isotonic. So when we have too much water moving into a cell, we can get something called cytolysis. So this is where, lysis means to burst, so this means that the cytoplasm basically bursts. So this means that each cell gets really fat, and then eventually the membrane can't contain the cell anymore and the cell will burst. So this is obviously very bad for the cells. On the other side, if water moves out of the cell can become shriveled, there's not much cytoplasm left in the cell and they can't perform their normal functions. And in a plant cell, what this will do is it will mean that the cell membrane actually pulls away from the cell wall. So if you don't remember the structure of a plant cell, remember to go back to the cells module. If the cell membrane pulls away from the cell wall of a plant, we call this plasmolysis. So ideally, for a cell to function optimally, it needs to be in an isotonic solution, because otherwise you're going to either get the cells becoming shriveled or the cells exploding because they've got too big. Okay, so we've now talked about diffusion and specifically osmosis, but now I'm going to talk a bit about facilitated diffusion. So some substances can't just diffuse through the phospholipid bilayer as they are. So we have things like large polar molecules, which include amino acids and glucose, and we also have charged molecules like ions. And as I said earlier, these can't just easily diffuse through the cell membrane. So these require facilitated diffusion. And this is done by two things. This is done by channel proteins or carrier proteins. So channel proteins are basically like a tunnel through the membrane. So it's hydrophilic, and things can just travel right through the channel, and they go from areas of high to low concentration. So molecules will just diffuse and use this as a method to get through the membrane. However, a lot of the time these channels are quite specific. And they can also be gated, which means that only certain things can go through the channel. And this allows the cell to control what goes in and out of the cell. Then we also have these things called carrier proteins. And I said that the channel protein is like a tunnel and it has a very fixed shape, but the carrier proteins actually normally have two different conformations. And what happens is once a molecule binds to the binding site in the carrier protein, it will change shape, and then it will allow the molecule to go through to the other side of the membrane. So the thing to remember here is that although these things do require additional proteins to help the diffusion happen, this is still diffusion. So it is still going from areas of higher concentration to areas of lower concentration. So the next thing I'm going to talk about is what happens when we want to get something from an area of low concentration to an area of high concentration. And we call this active transport. So active transport is the movement of molecules or ions across a cell membrane from a region of lower concentration to a region of higher concentration against the concentration gradient, and this uses energy. So really important thing to remember here is that you need energy to have active transport. Active transport goes against the concentration gradient, and because of that, you need to use energy to push those molecules in the opposite direction to what they would naturally want to do. So there are a couple of ways that this can happen. So we also can have carrier proteins as before, and these just use ATP, so they use the energy of the cell to change conformation, and they can move molecules from areas of lower concentration to areas of higher concentration. We also have another form of active transport, which is called cotransportation. So with cotransportation, we have the movement of two molecules, one via active transport and one via diffusion. And these use carrier proteins, and we call these cotransporters. And what happens is, instead of a molecule going through on its own, the molecule goes through with another molecule. So, for example, you might have glucose and sodium. When the sodium moves from an area of high concentration to low concentration, its movement through the cotransporter allows the movement of glucose with it. And this can either be in the same direction or in the opposite direction. If it's in the same direction, we call that a symporter. And if it's in the opposite direction, we call that an antiporter. Basically, it's like the glucose is piggybacking on the movement of the sodium. So this normally happens with ions, and one thing will be moving with the concentration gradient from area of high to low concentration, and this allows the carrier protein to pump a secondary molecule in the opposite direction, so from areas of low concentration to high concentration. I understand that this is quite confusing, so just keep going over this, and if you have any questions, please do let us know. Okay, so I think that is pretty much everything now on the membrane side of this lesson. So then we're going to now go on to surface area to volume ratio. So surface area to volume ratio is going to come up time and time again in this module, but also in all of biology. So it's really important that you understand this before moving forward. So surface area to volume ratio, right, has two parts to it. So there's the surface area and volume. So surface area, you will probably know from maths, is the outside area of an object, and it's the surface that is exposed to the external environment. Volume is the amount of space that an object occupies. You know, if we think about a cube has 6 sides, so that would be its surface area, and its volume would be how big that actual cube is. So that would be the surface area of one side cube. That is how we work out the volume of a cube. The important thing to know is that volume increases much more rapidly than surface area as an object increases. So the volume of an object will get much bigger, but the surface area will only increase slightly. And this might sound a bit confusing, so I'm going to go on to exactly some examples of this and exactly how that works in a second. So this is where this lesson is going to get a bit more mathsy. So there's certain shapes that you will need to know how to calculate their surface area and also their volume for this module. So we're going to start easy. We've got a cube. So to work out the surface area of a cube, what you would do is you work out the surface area of one side of the cube. So that would be the height times the height, because the height and the width of the cube are going to be the same. And then you would times that number by 6 because you have 6 sides on the cube. However, to work out the volume, what you would want to do is you have to do the height times the length times the width. So in this case, they are all the same. So that would be whatever number that is cubed. So times itself 3 times. So I'm just going to give an example here to help that make a bit more sense. So if we have a cube that has a height of 2, one side will have a surface area of 4, as it will be 2 times 2, which gives you 4. And then we would times that number by 6 to get the total surface area. So 6 times 4 is 24. The area is 24, but then if we want to work out the volume, we would have to do 4 times 4 times 4. So 4 times 4 is 16, and then if we times that by 4 again, we get 52. So 52 centimetres cubed would be how much volume that cube occupies. With other shapes, we need to use some other calculations. So if we have a cuboid, it's very similar, but the height and the width are not necessarily the same, and also the length can be different. So for the volume of a cube, it is height times width times length. And to work out the surface area, you have to work out the surface area of each of the faces and then add them together. Then we also need to know how to work out the volume and the surface area of a cylinder. So for the volume of a cylinder, it is pi r squared times height. So r is the radius of the circular face. And then for the surface area, you have to work out the area of the two circular ends and then also the middle. So the way that we do this is 2 pi r squared plus 2 pi r times height. This all sounds very confusing, but just practice and write down all these formula and it will eventually go in. So we're now going to just go over an example of how, as I said before, as size increases, the volume increases much more rapidly than the surface area. That kind of sounds quite abstract, so I'm going to give you some examples here and you will see how this works. So if we have a cube that is 1 centimetre by 1 centimetre, the surface area would be 6 centimetres squared and the volume would be 1 centimetre cubed. Yep. So we have a surface area to volume ratio of 6 to 1. If we have a 2 centimetre cube, then the surface area would then be 24 centimetres squared and the volume would be 8 centimetres cubed. So in this case, the surface area to volume ratio would be 3 to 1. So you can see that the surface area to volume ratio has decreased because the surface area has not increased as much as the volume. Then if we have a cube that is 3 centimetres, it would be, the surface area of that cube would be 54 centimetres squared and the volume would be 27 centimetres cubed. In this case, the surface area to volume ratio is then 2 to 1. So you can see that from the initial cube, which had a surface area to volume ratio of 6 to 1, to the bigger cube, there's now only a 2 to 1 surface area to volume ratio. As a cube gets bigger, its surface area to volume ratio decreases, and this also applies to organisms. So the larger an organism, the lower its surface area to volume ratio will be. Now, this is really important when we think about how exchange happens, because as we spoke about previously, different things need to come in and out of the cells, and also thinking more broadly, within a human or an animal or a plant, we have a whole system of things where things need to go in and out. So, we breathe, we eat, all these things. We need to get things in and we need to get things out. And surface area to volume ratio really affects this process. So single cell organisms like bacteria have a very high surface area to volume ratio because they're very small. However, more complex multicellular organisms, as they get bigger, they have a lower surface area to volume ratio. The larger the volume also means that the diffusion distance to get into the cells and into the tissue of an organism is bigger. So it will take longer for oxygen to get into a human cell than it would into a bacterial cell. This really impacts how organisms can function, and exchange is a vital part of life. So multicellular organisms have developed lots of different adaptations to facilitate this exchange by maximising their surface area to volume ratio. The rest of the module will keep coming back to this idea of surface area to volume ratio and how we can maximise that to have the most efficient exchange possible. So I'm just going to now go on specifically to a bit about gas exchange. In general, effective exchange surfaces in organisms will have a large surface area, which we discussed earlier, why that's useful. They will have a short diffusion distance, and they will maintain a high concentration gradient. Again, this is all things I mentioned earlier that improve the rate of diffusion. So by having these features in an exchange surface, this makes sure that the organism is going to have the maximal rate of diffusion happening. So in the next couple of lessons, Amelia's going to go over how gas exchange happens in humans, and I will also be going over how it works in plants, but I'm just going to give a bit of background on it now in a general sense, and then you'll get more specific in the future lessons. So as you know, exchange of gases from the atmosphere is really essential for life. So for humans, we need oxygen to respire, and plants require carbon dioxide to photosynthesize. And also then we need to get rid of our waste products, for example. So for respiration, we need to get rid of the carbon dioxide produced via respiration. And we also need to get in water into our cells. Not only is gas exchange really important for organisms, but we also need to have water, and water is vital for many functions within an organism. There needs to be a balance between water retention and gas exchange, and they are opposing. So the better your gas exchange, the more water you're going to lose, and the better you are at retaining water, the worse your gas exchange will be. Because of this, organisms have adapted many different ways to reduce water loss without negatively affecting gas exchange. So there's lots of different compromises that have been made that allow for sufficient supply of both oxygen and water to an organism. So there's two examples of this that you need to know. So there are terrestrial insects and xerophytic plants. So I'm just going to quickly look at terrestrial insects. So basically, small insects that live on the ground, like a grasshopper, for example, are always surrounded by air, and that means they're prone to drying out. They're prone to losing water very easily. The way that the insect has developed to prevent this is they are covered in this waxy coating, which is called an exoskeleton, and it prevents water being lost through their surface. However, this also makes gas exchange very difficult. So if they didn't have this, gas exchange would be very easy, but because of this, it means that it's harder to get gas in and out of the insect. So because of this, they have evolved a different breathing system, which we call the tracheal system, which is made up of many tiny tubes that carry oxygen directly to all the tissues in the body. There are lots of tiny openings in the exoskeleton where gas can come into the tracheal system and then be distributed throughout the body. So this is a way that these insects have evolved in order to maximise their gas exchange without losing too much water. Okay, and then if we look at xerophytic plants, these are plants that live in conditions where there's very limited fresh water. So they have lots of different adaptations to conserve water while still allowing for gas exchange. So you will, if you think about a cactus, you will be able to think of a lot of these. So they have very few stomata. Again, go back to the cells module if you don't know what a stomata is. They have very sunken stomata. They have hairs surrounding the stomata, and these protect water from being lost through the stomata when they're open. They also have very small or needle-shaped leaves, which reduces the water loss, and they also have a waxy cuticle that prevents water from evaporating from the surface of the leaves. And 2 examples of this type of plant are cacti and marram grass. Okay, so I think that is everything for today. I know that was quite a lot of information. Really do take the time to go back, re-listen, and check that you do get everything before you move on to the next few lessons, because this is really going to be important going forward. So what we went over today was the fluid mosaic model and how transport proteins are used to move substances in and out of cells. And we looked at surface area to volume ratio and how that can affect the rate of exchange in an organism. And then we briefly looked specifically at gas exchange and some adaptations that organisms have made to minimise water loss whilst maintaining high gas exchange. So like I said earlier, going forward, the next part of this module is going to be looking specifically at human exchange systems, and then we will be looking at plant exchange systems. So
thank you so much for listening. Remember, you can access additional content on our Patreon page by searching for Equity Tutors, where we have a second 30-minute lesson every week, plus monthly bonus content. You can also find us on most social media platforms. We will keep you updated on new content, and you can find us there by searching for Equity Tutors UK. Please like, share, subscribe and comment wherever you are listening, and if you're enjoying, please leave a review. Bye!
bye!